Hello, everybody, and welcome back. We would like to start the next session, which is on Japanese encephalitis. Dr. Walter, can you go ahead and get started, please? Our work group lead.
Previously, this has been called the Flavy Vaccine Working Group. At one time, we actually were just the JE vaccine group, but we combined to be the Flavy Vaccine Working Group, and now we have split back up into two work groups again, into the Japanese encephalitis work group and the dengue vaccine work group. The reason being is that the amount of work to be done and the expertise needed for both work groups, so we decided to split the work groups up. So the Japanese encephalitis work group will continue working on finishing the updated Japanese encephalitis vaccine recommendations, and we're expected to complete our work hopefully in February 2019, as you'll shortly hear. And then the dengue vaccine work group will be chaired by Dr. Robert Atnar, who is also on the Japanese encephalitis vaccine work group. And he will help to present at the next meeting considering the recommendations for possible use of a dengue vaccine in the United States and present the terms of reference and background information for the formation of the work group at the February meeting. So stay tuned for that. This is just a list of our Japanese encephalitis work group members. I'd like to point out here our CDC lead, Susan Hills, who will be presenting today's session. So the objectives in terms of reference for our group are to review the newly available safety and immunogenicity data for an activated varicella culture-derived JE vaccine, or JEVC, as Susan will really refer to this during the rest of the presentation today. We have reviewed the epidemiology and risk of Japanese encephalitis in travelers, and we are reviewing the ACIP recommendations for the use of Japanese encephalitis vaccine in consideration of all the updated data that we've heard about and that you all have been hearing about. We will then update the MMWR recommendations and report. So this work group started its work in 2015. We are not fast moving. So we have made a number of presentations to the ACIP since 2015. We got detoured a little bit by a little Zika outbreak. So people got pulled off duty to other duties for a while, but we have reconvened. So since 2015, there have been several presentations which are listed here. Some of these presentations date back to 2015, so we're going to be updating some of them today. Early on, we presented the immunogenicity, JEVC, in adults over 65 years of age. The information about the accelerated dosing schedule at 0 and 7 days was presented. Duration of protection was presented. Information has been presented about a booster dose for children under age 17 years of age. At more recent meetings, we've updated information and presented it to ACIP about the epidemiology and risk of Japanese encephalitis in U.S. travelers. We recently heard some post-marketing safety and adverse event data. At the last meeting, there was a comparative analysis of vaccination strategies was presented, and also a grade analysis of JE vaccine for U.S. travelers. So for today's session, you'll hear several things. We will be presenting the evidence to recommendations framework for the updated JE vaccine recommendations. Susan will be presenting that. She will review earlier sessions from back almost 2015 on the accelerated dosing schedule for data in adults. She'll review the booster dose recommendations, which strengthen the current permissive recommendation and expanded recommendations to include children under 17 years of age. And then we anticipate at the next meeting, we'll take a final vote at the February 2019 meeting on the recommendations. Susan?
Thank you, Dr. Walter, and good afternoon, everyone. My first presentation today, which I'm presenting on behalf of the Japanese Encephalitis Work Group, is the Evidence to Recommendations Framework for Japanese Encephalitis Vaccine. I'll be discussing the various components of the ETR framework, which you've already seen in presentations this morning as well. The overarching policy question that the work group addressed was, should JEVC be recommended for use in persons aged 2 months and older at risk of travel-related exposure to JE virus? The components of the PICO question were the population, which is persons aged 2 months and older travelling to risk areas in Asia, the intervention, which is JEVC administered as a 2-dose primary series, the comparison, which is no JE vaccine recommended, and the critical outcomes, which were short and long-term seroprotection, serious adverse events, and adverse events of special interest. The following slides will provide some background information that's important for understanding the JE vaccine recommendations. JE is caused by a mosquito-borne flavivirus. It occurs in most of Asia and parts of the Western Pacific and is the leading vaccine preventable cause of encephalitis in Asia. Most JE virus infections in humans are asymptomatic, with fewer than 1% of infected people developing neurological disease. However, when disease does occur, it's often severe. Overall, about 20 to 30% of patients die, and 30 to 50% of survivors have significant neurologic, cognitive or behavioural sequelae. There's no specific antiviral therapy, and so treatment consists of supportive care. Currently, even with national vaccination programs in some endemic countries, there still are an estimated 68,000 JE cases annually in Asia, with an overall incidence for all age groups of approximately 1.8 cases per 100,000 population. The highest risk for infection is in rural agricultural areas because the primary breeding site for the main vector is rice fields. For most travellers to Asia, the risk for JE is very low, but varies based on travel destination, duration, season, activities and accommodations. A JE vaccine was first licensed in the United States in 1992, and in the 25 years from 1992 to 2017, only 12 JE cases were identified among US travellers or expatriates. Travellers with longer trips or increased rural and outdoor exposures are at higher risk of acquiring JE virus infection. Among the 12 US traveller cases, 8 had travelled for a month or longer. 3 cases travelled for less than a month but spent at least one night in a rural area, and 1 case travelled for less than 1 month but we have no information on their itinerary or activities. JEVC, manufactured by Valneva as Ixiaro, is the only JE vaccine currently licensed and available in the United States. The vaccine was licensed for adults aged 17 years and older in 2009, and the licensure was subsequently extended to children aged 2 months through 16 years in 2013. The primary series is 2 doses administered 28 days apart, and the vaccine costs approximately $600 for the 2-dose primary series. There are no efficacy data for Ixiaro. Because of the availability of several effective JE vaccines in Asia, randomised controlled efficacy trials to evaluate a new JE vaccine would have been logistically difficult and also potentially unethical. However, there is an established immunologic correlative protection, and that is a JE virus 50% plaque reduction neutralisation test, or PRNT 50, of greater than or equal to 10. The vaccine was licensed based on a non-inferior neutralising antibody response compared with a licensed mouse brain-derived JE vaccine. Following licensure of JEVC for adults in 2009, ACIP approved recommendations for use of JE vaccine in US travellers. And then in 2013, following licensure of JEVC for children, a grade analysis was performed and the JE vaccine recommendations were reviewed, but no changes were made. The current review of the JE vaccine recommendations is a routine review in consideration of new safety, immunogenicity and traveller risk data. In addition, an updated MMWR recommendations and reports document will be published that will combine information from the existing 2010 MMWR document and policy notes that have been published since that time, as well as incorporating new indications and dosing schedules. The first specific domain of the ETR framework was public health importance, and I'll present now the workgroup considerations and conclusions. JE virus is the leading vaccine-preventable cause of encephalitis in Asia and parts of the Western Pacific. In the highest risk areas in Asia prior to vaccination programs, incidence rates as high as about 20 per 100,000 children per year were reported. So JE is clearly of public health importance in the region. However, based on number of cases, JE cannot be considered a substantial public health problem for US travellers. Based on 12 reported JE cases over a 25-year period and 4 to 5 million US trips to Asia annually during this period, the estimated risk is less than 1 case per million trips to Asia. There is no risk of subsequent local transmission in the US from an infected traveller because of low-level viremia when humans are infected with JE virus. While JE can't be considered a public health problem for US travellers in general, it is an important individual concern for some travellers. For some persons, like those taking up long-term residence in rural areas of Asia, risk might approach a similar level to populations of endemic areas. Additionally, when disease occurs, only supportive treatment is available and the outcome is often severe, with a high rate of death and disability following clinical disease. So the workgroup concluded that firstly, JE is clearly a public health problem in endemic countries. Secondly, for US travellers, the question of public health importance is really not directly applicable, as it's an individual traveller concern. The JE vaccine is typically paid for out of pocket, so the question is also less relevant than for many other vaccines. Finally, for a judgment within the ETR framework, the workgroup decided the public health importance varies related to the individual person's risk based on their itinerary and activities. For some travellers, risk of a potentially severe disease is sufficiently high that vaccination should be recommended. The next domain was benefits and harms. The workgroup presented the grade analysis to ACIP in June 2018, so I'm just going to briefly summarise the benefits and harms based on that analysis. Data to assess seroprotection were from 4 randomised controlled trials and 8 observational studies. Seroprotection rates after the 2-dose primary series in all studies in individuals up to 64 years of age were at least 95% at 1 month and 83% or higher at 5 to 6 months. When data from randomised controlled trials were combined and weighted using a random effects model, seroprotection rates after JEVC compared to rates in recipients of a JE vaccine previously licensed in the United States or JE vaccines licensed in other countries were non-inferior at 1 month and significantly higher at 5 to 6 months. The workgroup therefore concluded the vaccine benefits are large. In randomised trials, there were no significant differences compared to placebo or other vaccines for serious adverse events or adverse events of special interest, including fever, rash, neurological, hypersensitivity or medically attended events. There were no safety signals in post-marketing surveillance and no patterns in the timing or types of adverse events. So the workgroup concluded that the vaccine risks are small. The level of evidence for the grade analysis benefits and risks was type 1 or travellers but has potentially serious outcomes, and that JEVC has no important safety concerns but rare serious adverse events can occur. The workgroup felt the benefits outweigh the risks for some travellers. Healthcare providers should discuss a traveller's risk based on their itinerary and activities, and vaccines should be targeted to travellers at increased risk. The target population perception of value was the next domain that was considered by the workgroup. The questions we considered were, does the target population feel that desirable effects are large relative to undesirable effects? And is there important uncertainty or variability in how much people value the main outcome? To address these questions, a population survey was conducted using the Porter-Novelli survey mechanism. The survey population included randomly recruited participants representative of the non-institutionalised US population, and overall there were 6,427 respondents. Participants were presented a scenario on travelling overseas and provided estimates of disease risk and potential outcomes for a disease such as JE, and vaccine safety, risk of side effects and cost. They were queried regarding the likelihood of receiving the vaccine and factors important in their decision. The results showed that in regard to the likelihood of receiving the vaccine, 32% said they were very or somewhat likely to receive the vaccine, 43% said they were very or somewhat unlikely to receive the vaccine, and 25% were unsure. For those who were very or somewhat likely to receive the vaccine, the most influential factors were disease risk and risk of severe disease outcomes. For those who responded they were very or somewhat unlikely to receive the vaccine, influential factors were vaccine cost and also disease risk. The workgroup concluded that there was variability in the population's perception of the potential benefits and harms of vaccination for a rare but potentially severe disease. Some of the population clearly value the availability of a vaccine to prevent a potentially serious disease, but others are less likely to place value on an expensive vaccine, which, while safe, has the possibility of rare serious side effects. With the risk of disease being considered a reason by some to receive the vaccine and by others to not receive the vaccine, there's clearly substantial variability in how people perceive and tolerate risk. Acceptability to key stakeholders was the next domain addressed by the workgroup, which we considered in the context of acceptability of the proposed JE vaccine recommendations. Travel medicine providers were considered an important stakeholder. Mechanisms to survey US travel medicine practitioners were investigated, but none could be identified within the timeframe available. However, 4 members of our ACIP workgroup are practising travel medicine practitioners and members of the International Society of Travel Medicine, and they played an active role in discussions about the recommendations. We also reviewed opinions about JE vaccine recommendations expressed in publications and in letters submitted to ACIP by US healthcare providers, and we had 2 practitioners with dissenting opinions on the recommendations present to the workgroup. Overall, the workgroup concluded that there is stakeholder consensus that there is a low risk for most travellers, there's a need to inform travellers about disease risks and prevention measures, and that vaccination should be targeted to travellers at higher risk. The workgroup felt that the recommendations would probably be acceptable to most stakeholders as they're based on individual clinical decision-making with consideration of several important factors listed here, which include the risks related to the specific itinerary, the likelihood of further travel to JE endemic countries, the high morbidity and mortality of JE when it occurs, the possibility but low probability of serious adverse events, and the traveller's personal perception and tolerance of risk. Resource use was the next domain considered by the workgroup. And the ETR question we addressed was, is the intervention a reasonable and efficient allocation of resources? Again, the workgroup considered this question in the context of the proposed JE vaccine recommendations that would target higher risk travellers. A comparative analysis of strategies was presented to ACIP in June 2018 by CDC's Health Economics and Modelling Unit, with the objective being to provide perspective on numbers needed to vaccinate and cost per case averted for travellers with different itineraries and disease risk, and the cost implications of possibly expanding the current JE vaccine recommendations to a broader group of travellers. Before summarising the key results, I'll remind you of the groups that were included in the analysis. Risk Group 1 included travellers who plan to spend a month or longer in JE endemic areas and approximates a group for whom JE vaccine is recommended. Risk Group 2 includes travellers who will spend less than a month in JE endemic areas, with at least 20% of their time doing outdoor activities in rural areas. And this group approximate travellers for whom JE vaccination should be considered after evaluating their itinerary and weighing the benefits, risks and costs. And Risk Group 3 includes a remainder of shorter term and lower risk US travellers to Asia for whom JE vaccine is not recommended. This table shows the estimated proportion and number of travellers to Asia per year in each of the risk groups. Risk Group 1 represents about 20% of all travellers, or about 0.9 million travellers per year. Risk Group 2 represents about 25% of travellers, or about 1.2 million travellers per year, and Risk Group 3 represents the remaining 55% of travellers, or about 2.7 million travellers per year. The first key finding from the analysis was that the numbers needed to vaccinate to prevent a case are presented in this table, and to prevent one case of JE among US travellers under the base case scenario, which is shown in the middle column, the number of travellers who would need to be vaccinated was 0.7 million in the highest risk group, Risk Group 1, 1.6 million in Risk Group 2 and 9.8 million in Risk Group 3. Because baseline incidence is based on reported cases, and to address any uncertainty about the sensitivity of surveillance, we also did a sensitivity analysis using an incidence 100 times higher, even though we would not expect 100 cases to be occurring for every one case reported. As shown in the final column, with incidence 100 times higher, the numbers needed to vaccinate to prevent a case were 7,000, 16,000 and 98,000 in the three risk groups respectively. The cost to prevent one case of JE in travellers is shown on this slide. With baseline assumptions, the cost from a societal perspective to prevent one case in the highest risk category is about $0.6 billion. Comparatively, for those in Risk Group 2, the cost per case averted is $1.3 billion, and for those in Risk Group 3, it's $7.9 billion. With incidence increased 100 times in the sensitivity analysis in the column on the far right, the cost per case averted in each of the three risk groups is $5 million, $12 million and $78 million respectively. This slide shows the additional cost to prevent an additional JE case by expanding the JE vaccine recommendations. If JE vaccine recommendations were expanded from Risk Group 1 to routinely recommend vaccination for all travellers in Risk Groups 1 and 2, it would cost the society an additional $1.6 billion to prevent one additional case of JE. Similarly, expanding JE vaccine recommendations from Risk Groups 1 and 2 to all travellers will cost the society an additional $14.6 billion to prevent an additional JE case. There were two general resource considerations that the workgroup noted when considering this domain. Firstly, compared to vaccines used universally in the population, there are a relatively small number of travellers to Asia with an itinerary that puts them at increased risk for JE. Secondly, travel vaccines are usually paid for by the travellers themselves and are not covered under the Vaccines for Children program or by most private insurers. So overall, the workgroup conclusions were that JE vaccine is probably not an efficient use of societal resources, as it's an expensive vaccine for a low-risk disease. However, the question of resource use is less applicable to a travel vaccine, as it's typically paid for by individuals, and they are deciding on purchase of a vaccine to prevent a potentially severe disease. The comparative analysis supported the proposed tiered JE vaccine recommendations, as it indicated a large increased cost to society to prevent a case of JE when risk groups 1 and 2 were routinely included in recommendations compared with risk group 1 alone, supporting a more cautious approach or consideration of vaccination for those in risk group 2. In addition, there was a very large increased cost to society if risk group 3 was included, which does not support a broad recommendation for JE vaccination for all travellers. Finally, vaccine recommendations targeted to higher risk groups probably a reasonable allocation of resources, as the financial implications related to vaccine purchase will be borne by the travellers most at risk of a severe disease. The next domain was feasibility, and the work group again considered this in the context of implementation of proposed risk-based recommendations. Administration of vaccine would be feasible during a pre-travel consultation. With risk-based recommendations, barriers to implementation might include lack of understanding by healthcare practitioners of factors that might increase the risk for JE. However, specific information is provided in a table accompanying the recommendations, and other resources, such as the CDC Yellow Book, are readily available to practitioners. Therefore, the work group considered risk-based recommendations were probably feasible to implement. Next, the work group considered balance of consequences, and considered in the context of recommendations targeting higher risk travellers, the work group felt the desirable consequences would probably outweigh the undesirable consequences in most settings. So I'm now going to discuss the specific JE vaccine recommendations proposed by the work group. Based on the vaccine data and additional factors considered within the evidence to recommendations framework, the work group felt there was sufficient information to move forward with updated recommendations, and that vaccination should be recommended for individuals based on individual clinical decision making. The work group also considered only minor updates were needed to the existing recommendations for use of JE vaccine in travellers. ACIP members should have a copy of the recommendations in their folders, and they lay out the recommendations in the way they would be formatted, and they include information that will be included in an additional box. In the next 5 slides, though, I'm going to present the key information, and I'm presenting all this information because the work group considered all the components are important as part of the recommendations. So the text of the proposed JE vaccine recommendations is as follows. JE is a very low-risk disease for most travellers to JE endemic countries. Some travellers will be at increased risk of JE virus infection based on their planned itinerary, and factors that increase the risk of JE virus exposure include longer duration of travel, travel during the JE virus transmission season, spending time in rural areas, participating in extensive outdoor activities, and staying in accommodations without air conditioning, screens or bed nets. Box 1 provides additional information on these factors, and as I mentioned, were included in the background information that ACIP members received. Continuing with the text of the recommendations, healthcare providers should assess a traveller's risk for mosquito exposure and JE virus infection based on their planned itinerary and discuss ways to reduce their risk. All travellers to JE endemic areas should be advised of the importance of taking precautions to avoid mosquito bites to reduce the risk for JE and other vector-borne diseases. These precautions include using insect repellent, permethrin-impregnated clothing and bed nets, and staying in accommodations with screened or air-conditioned rooms. For some people who might be at increased risk for JE based on travel duration, season, location, activities and accommodations, JE vaccine can further reduce the risk for infection. The decision whether to vaccinate should be individualised and weigh the risks related to the specific travel itinerary, likelihood of further travel to JE endemic countries, high morbidity and mortality of JE when it occurs, availability of an effective vaccine, possibility but low probability of serious adverse events following vaccination, and the traveller's personal perception and tolerance of risk. JE vaccine is recommended for persons moving to a JE endemic country to take up residence. Longer term, for example, 1 month or longer travellers to JE endemic areas, and recurrent travellers to JE endemic areas. JE vaccine also should be considered for shorter term, for example, less than 1 month travellers, with an increased risk of JE based on planned travel duration, season, location, activities and accommodations. Vaccination also should be considered for travellers to endemic areas who are uncertain of specific duration of travel, destinations or activities. JE vaccine is not recommended for travellers with very low risk itineraries, such as urban travel only for a short duration or travel outside of a well-defined JE virus transmission season. The minor changes from the current JE vaccine recommendations are that there is additional information on factors that increase JE risk to assist providers with decision making. There is longer term travel is no longer defined as a specific cutoff of 1 month or longer. The consideration of vaccination for travellers to an area with an ongoing JE outbreak has been removed, and there are small wording changes to address questions that have been raised, including changing expatriates to persons moving to a JE endemic country to take up residence. The second topic for presentation today is the accelerated schedule for JEVC for adults aged 18 to 65 years. These data were presented to ACIP previously, as Dr. Walter mentioned, but as we would like ACIP members to vote on the issue in February 2019, my presentation today is going to briefly review and summarise these data. As a reminder of the timeline of relevant events in regards to this topic, in March 2009, FDA approved JEVC for use as a 2-dose primary series administered in the standard 0 and 28-day schedule. In October 2015, the manufacturer presented data to ACIP for an alternate accelerated primary series at 0 and 7 days in adults, and ACIP noted that the data looked promising. Then in December 2017, the manufacturer submitted the BLA amendment to FDA, and in October 2018, FDA approved the accelerated primary series. The data supporting the accelerated schedule came from one randomised trial among adults aged 18 to 65 years, and the study was conducted at 7 sites in Europe. The study had 4 arms, including JEVC with rabies vaccine in an accelerated or standard schedule, and JEVC and rabies vaccine administered alone in standard schedules. There was no arm that included JEVC administered alone in accelerated schedule, so the data I'm going to present are the comparative data for JEVC administered with rabies vaccine in an accelerated or a standard schedule. This table shows seroprotection rates and geometric mean titers or GMTs at 28 days after the second JEVC dose of the primary series for the accelerated schedule, which is in the middle column, and the standard schedule, which is in the final column. Seroprotection rates are shown in the top row, and at 28 days were 99% for the accelerated schedule and 100% for the standard schedule. GMTs, shown in the bottom row, were significantly higher for the accelerated schedule compared with the standard schedule, with GMTs of 690 and 299 in the 2 groups respectively. This slide shows seroprotection rates and GMTs at approximately 1 year, and seroprotection rates were 94% for the accelerated schedule group and 86% for the standard schedule group. GMTs were again significantly higher for the accelerated schedule group compared with the standard schedule group, with GMTs of 117 and 39 for the 2 groups respectively. This slide shows solicited adverse events within 7 days after co-administered JEVC and rabies vaccine for the accelerated and standard schedule groups. There were no significant differences in the percentage of subjects with the systemic adverse events, which are shown above the line, or local adverse events, which are listed below the line. So the proposed wording for a new recommendation for an accelerated schedule would be, in adults aged 18 to 65 years, JEVC can be administered in an alternate primary schedule of 2 JEVC doses administered 7 days apart. The next topic in my presentation today is JEVC booster doses. These data have also been presented to ACIP previously. However, as a vote is again being requested in February 2019, I'm going to review and summarise these data. The timeline of events relevant to booster doses for JEVC is as follows. So in September 2010, FDA approved a JEVC booster dose for adults aged 17 years and older. In February 2016, the manufacturer presented data to ACIP for booster doses in children aged 14 months and older. And in June 2017, the manufacturer submitted to FDA a BLA amendment for a pediatric booster dose, and in April 2018, FDA approved the pediatric booster dose. The current ACIP recommendations for a JEVC booster dose are for adults aged 17 years and older. And the wording of the booster dose is here on the slide. If the primary series of JEVC was administered more than 1 year previously, a booster dose may be given before potential JE virus exposure. The topics for consideration today are lowering the recommended age for a booster dose to include children aged 14 months and older, and strengthening the current permissive booster dose recommendation. I'll start with a review of the data supporting a booster dose in children, and this comes from one open-label randomised trial among children aged 2 months through 17 years conducted in the Philippines, which is a JE endemic country. 300 children were randomised to receive or not receive a booster dose of JEVC. The 150 children in the booster dose group were administered a booster at 11 months after the second dose of the 2-dose primary JEVC series. And data from the children in this group were analysed by age group based on the vaccine dosage given, which is 0.25 mils for children aged 2 years and younger and 0.5 mils for older children. This slide shows seroprotection rates and GMTs at 11 months after a 2-dose primary series of JEVC or prior to the booster dose, and then at 1 month after the booster dose. These data are for 81 children aged 14 months to 2 years who received a 0.25 ml booster dose. Seroprotection rates shown in the middle column were 98% prior to the booster and 100% after the booster. GMTs, shown in the final column, did show a substantial increase, with a GMT of 67 prior to the booster and 2,911 after the booster. Here you can see the same result as the previous slide, but for children aged 3 to 17 years for whom the dose is 0.5 mils. So among 67 children in this age group, seroprotection rates were 90% prior to the booster and 100% at 1 month after the booster. And again, GMTs were significantly higher after the booster dose, with a GMT of 40 prior to the booster dose and 1,366 after the booster. This slide shows our data collected at 2 years after a booster for those children who received a booster dose, and comparative data for the same time point for those who did not receive a booster dose. So seroprotection rates at 2 years after the booster dose were 100% both for younger children who received the 0.25 ml booster and for the older children who received the 0.5 ml dose. Among children who did not receive a booster dose, the seroprotection rate was 90%. Comparing GMTs in children who received a booster dose with those who did not, GMTs were 4 to 7-fold higher in those who received a booster dose. Safety data were collected for 2 years after the booster dose. Solicited adverse reactions were reported less frequently following the booster dose than following the primary series. And the most common solicited adverse reaction following the booster dose was fever, which occurred in 8% of children. So based on the apparent enhanced durability of the antibody response in children elicited by the administration of a booster dose, the work group concluded that the current booster dose recommendation for adults should be modified to include children 14 months and older. Both based on the lower standard dose of JEVC for younger children aged 14 months to 2 years should receive a single 0.25 ml booster dose, and children aged 3 years and older should receive a 0.5 ml booster dose. The final data I'd like to present today are the data that support strengthening the current permissive booster dose recommendation. Data supporting the current booster dose recommendation came from 3 clinical trials that provided data on persistence of a protective neutralising antibody response at 12 to 15 months after a primary 2-dose series of JEVC in adults. And these studies were conducted in Austria, Germany, Northern Ireland and Romania. In the middle column, you can see seroprotection rates at 12 to 15 months after the 2-dose primary series of JEVC, and rates were quite variable in the 3 studies, ranging from 58 to 83%. GMTs in the final column range from 18 to 41. As I mentioned, these studies were conducted in Europe, where tick-borne encephalitis or TBE vaccine is available in some of the countries. And TBE is a flavivirus related to JE virus, and there was concern that there might have been a boosting effect of TBE vaccine, which could explain some of the variability in these results. So as a result, the manufacturer conducted a post hoc analysis that stratified subjects by TBE vaccination status, and these data were presented to ACIP in February 2016. This slide shows the results from this post hoc analysis. And the 2 groups include subjects who had not received TBE vaccine, with the data shown in the middle column, and subjects who had received TBE vaccine either prior to or after JE vaccination, and those data are shown in the far right column. The 3 rows show seroprotection rates at 1 year, 3 years and 5 years after the primary JEVC series. The seroprotection rates at all 3 time points were lower for those who had not received TBE vaccine compared with those who had received the TBE vaccine. The work group concluded that after a 2-dose primary series, long-term JE seroprotection rates are lower in those not administered TBE vaccine compared with those who are administered TBE vaccine. TBE vaccine is not available in the United States, as you know, and other flavivirus vaccines, such as yellow fever vaccine, are not routinely administered with JEVC. Therefore, among US travellers, duration of protection following a booster dose of JEVC is likely to be most similar to the subjects not administered TBE vaccine, who had lower seroprotection rates through the 5 years. So based on these data, the work group recommended the permissive booster dose recommendation should be strengthened. So summarising all these booster dose data that I presented today, the proposed new recommendation for JEVC booster doses is shown on this slide, with the relevant changes underlined. So for adults and children aged 14 months and older, if the primary series of JEVC was administered more than 1 year previously, a booster dose should be given before potential JE virus exposure. Children aged 14 months to 2 years should receive a single 0.25 ml booster dose, and adults and children aged 3 years and older should receive a single 0.5 ml booster dose. I'll complete my presentation today with information on the next steps for our JE vaccine work group. So to complete the JE vaccine workgroup activities, we request ACIP members vote on the 3 topics that I presented today. And as a reminder, these are minor updates to JE vaccine recommendations for US travellers, an alternate accelerated vaccination schedule, and for the booster dose recommendation, minor modifications to strengthen the wording and lowering the recommended age to include children. The workgroup plans to provide a draft updated MMWR recommendations and reports to ACIP prior to the February meeting. Thank you.
Great. Thank you so much, Dr. Hills. We can start with questions and comments. It looks like Dr. Romero has a comment.
So let me begin by saying congratulations to you on an extremely lucid presentation. This was very, very clear, and I guess the question I have is why aren't we voting on it today? But I know the answer. And congratulations to the work group for distilling all this information down. So now my question, and excuse my density for not understanding this, but on the slide just before you present your JEVC accelerated schedule for adults aged 18 to 60 or 65 years on minor changes for current JE vaccination recommendations. So it says longer term travel no longer specifically defined as a cutoff of greater than or equal to 1 month. Are you now leaving this up to physician discretion?
We are still giving the example of greater than 1 month as the example of longer-term travel, and that is based on our US traveller data. So when we looked at the 12 cases of JE in US travellers, we found that 67% of all our travellers had travel that were travel was 30 days or more. The reason we moved the very specific wording is because people were literally taking that at face value, and if it was 29 days, saying not recommended, 30 days recommended. So we wanted to put across the message that it was longer term travel, but not be so definitive.
Thank you.
One other point to that, we didn't want people to be so focused that they neglected some of the other risk factors as well. So,
thanks, Chip.
Great. Dr. Bernstein?
I was wondering when the proposed new recommendations for the booster, if the getting it at the beginning is not necessarily a should, is there a reason that it's a should now, should be given, or rather than may be given?
So I think based on that analysis that looked at the TBE data and clearly lower seroprotection rates in those who hadn't received TBE vaccine, we didn't have those data available when we made the initial recommendation that said may be given. But looking at the two groups and those that had TBE compared to those who didn't have TBE vaccine, the lower seroprotection rates, we'd like to strengthen that in recognition of those data.
So then people who go repeatedly to Southeast Asia, so we would be letting them know that they should, if it's greater than a year after, they should come back for their booster.
If there's an ongoing risk of JE virus exposure, we would recommend a booster dose.
Thank you.
Okay, Dr. Moore?
Thank you. I really appreciate it. This one was challenging. And in looking at the wording of the recommendations, I had a quick question on JE vaccine being recommended for the extended travel to JE endemic areas. When you say areas, do you mean country or do you mean rural areas where there's ongoing transmission? I just was wondering which way it was intended by the work group to mean.
We intended it to mean areas. We didn't want to say countries, for example, because there's areas that are not endemic within countries. And we do have in the Yellow Book, for example, some information on areas within countries that are considered JE endemic areas. So it's travel to those areas or travellers who might be exposed within those areas.
Thanks. Because you do a really nice job of itemizing the things to factor into the decision to vaccinate, but then the very first statement is if you're gone for a month or longer or long-term travel, then you should do it. But then you come back to the last statement and say, it's not recommended if it's travel outside of a well-defined JE virus transmission season, which makes sense. So it could be the simplified recommendations could be a little bit confusing if it's travel for more than a month, but it's outside of a transmission season. What is the work group's intent?
Often those who are travelling for more than a month are going to span seasons, because those transmission seasons, if they're going overseas for that period, often will be both in the period when there may not be transmission and may be transmission. Certainly, if they're, again, it's an individual clinical decision making, so I'd say in that scenario to consider and discuss the various activities and what they're doing. We also need to keep in mind, so in some endemic areas there is year-round transmission, so that makes it easier, and then in others, it's a defined transmission season, so if they're at the very beginning, and they were going to stay in nice hotels, etc., may not be necessary. So I think it comes back to individual clinical decision making, but thank you for that.
Can I just
Dr.
clarify
Fisher?
that? Oh, Mark Fisher from Arboviral Disease Branch CDC. So for the longer-term travelers or recurrent travelers, we assume that they will, as Susan said, span the transmission season, or even if their primary travel will be to an urban area, they will spend time in rural areas. So we do still recommend the vaccine as the recommendation, say, for those longer-term or recurrent travelers, because we make the assumption that they're going to be at risk at some point during their travel.
Okay, thank you. I think for me, the high cost and the personal payment and the very, very low risk it's really important that the traveler understand before they invest $600 out of their pocket about what they're really, what their risks really are. And so I just want to make sure that those advising them have the best possible guidance.
Dr. Hunter?
I have a comment about how health inequities related to social economic factors might be an interesting issue with this, in that when you look at your conclusion that the benefits are large, you're talking about the individual who's paying $600. But the societal benefits are not necessarily huge because it's small numbers of people. But the kind of people that are probably going to be at higher risks are people who are going back to visit their relatives in a rural area and have very little money. And they medically are going to be at higher risk, but less able to pay. I don't see any way of dealing with this issue. I just wanted to bring it up and say that there's going to be inequities. And I think that a small comment at least should be said about that.
We did talk about that on the work group, Paul, and actually I raised that point because I used to do travel clinic, and I would have families with 3 kids come in, and they're facing quite a high expense. So you really weighed risks and benefits in those particular cases, and if they could afford the vaccine or not. And I think Elizabeth Barnett, who's on the group as well, has raised that issue at times.
Yes, thank you for that, Dr. Walter. We did have a very active discussion about that. And just for your awareness, we have made that comment in the additional information within the ETR framework. As you said, we couldn't see a way to deal with that within the recommendations, but it is noted. Clearly, I was presenting a summary here today.
Great. Dr. Kimberlin?
David Kimberlin, AAP Red Book. Forgive me for missing if it was stated. The Philippines study was enrolling, open label, enrolling 2 months of age to 17 years of age. Data were not presented from 2 months to 14 months, I don't believe. Well, I have a series of questions, but starting with that.
Right. So the primary series started at 2 months of age, and so those data have been presented previously. For the booster dose, that those who receive the vaccine at 2 months of age would be getting the booster vaccination, receive the primary series at 2 months of age, would be getting the vaccine at 14 months of age. So that's why the data for the booster dose were from 14 months through to 17 years of age. But there were children, younger children, starting off their primary series at that younger age. It's just when the booster dose is given that was presented here.
So if the recommendation is accepted by ACIP, the series would start at 2 months and the booster dose would be at 14 months.
Yeah.
I mean, as a lowest limit kind of thing.
That's right. So the vaccine is licensed and approved for children 2 months of age and older.
All right. And who? This is going to show some frustration I have, and I'm picking up on some of the comments I think made by the ACIP members as well. Who's kind of the target of this recommendation? The language is pretty mushy about who should actually get this. I'm not real clear that I think that putting an EG in front of the greater or equal to 1 month really means that it's not 1 month or longer. But that's the most sort of hard recommendation there is in there. And so I don't know exactly, I'm not sure, is this a travel medicine type of a recommendation, or is this somebody for the practicing pediatrician, for instance?
So the recommendations, we tried to base it as closely as possible on data. So what we do know is the first part of those recommendations are recommended for persons taking up residence in endemic countries. And then longer-term travellers, greater than 30 days. Again, it's based on the data we have available, and clearly, as you saw, there's very few cases, but we know that 67% of our traveller cases have occurred in about the 20% of all travellers who actually travel for that longer period. So there's clear evidence that is the highest risk group going to take up residence, travelling for longer than 30 days, or recurrent travellers, and so trying to focus on that longer term higher risk group as a recommendation with consideration of other factors in the second group.
Dr. Fisher?
Just to clarify also, Dr. Kimberlin, to answer your question, that the vaccine is, the recommendations are primarily targeted to travel health providers, some of which will be travel medicine physicians and other types of settings that provide recommendations in a broader context to travelers. JE vaccine, unlike yellow fever vaccine, can be administered in other places, and there are other, there may be some general pediatricians administering it, but it's really a vaccine that's being administered in the context of a pre-travel visit.
Dr. Welter?
Yeah, I just want to say, as a pediatrician that does general pediatrics who used to do travel medicine, we do not stock JE, Japanese encephalitis vaccine, in our clinic. We would refer a patient that we would want that consultation on to a travel clinic. So, and this really is meant for them, but it can be guidance for the general medical community as well.
Dr. Hahn?
Yeah, I just wanted to mention, I thought it was a little bit confusing, and I apologize because the slides went by, but the one slide where you look at the different by duration, and it said for travelers less than a month, here's some additional considerations like more than 20% of your time outside, and one of the additional considerations was duration of travel. I thought that was a little confusing because you've already put them into this less than a month bucket, kind of. So I don't know if you meant to distinguish between somebody going there for a week versus 3 weeks, like you might actually consider those two patients differently, but I just thought that could be a little bit confusing to people because they've already been told, okay, you're in this medium risk group or lower risk group based on less than a month. So
Right.
just a comment, I don't know how to make that any clearer, but if that was the intent to literally look at somebody traveling 1 week differently than 3 weeks, then maybe that's the best way to say it, but.
So thank you for that comment, and that was the intention, a 3-day traveller versus a 3-week traveller. And in the box that I didn't present, it does sort of provide information under all those headings, and duration is one of them to try and explain that more and what we mean. So I think that will clarify it, and thank you for that.
Great, thank you. We have one comment from the back of the room. Is that Dr. McIntyre?
Yes, Peter McIntyre again from Australia. I'm glad that the question of visiting friends and relatives came up. And it's something I wanted to check back with you about, because in the Australian context, I'm not sure if it applies in the US, there's fairly good information that the visiting friends and relatives group typically don't consult travel medicine providers, or uncommonly do. They would typically rely on primary care providers, often of their own ethnic group. And then the economic barrier is also relevant in the sense that, I'm not sure if this came up in the working group, but there's a Chinese manufactured live attenuated vaccine, which is on the national immunisation program of many countries in South Asia and East Asia, which is WHO pre-qualified. It's has an extensive safety record. It's been shown to be effective. It's a single dose product, and I can guarantee it would be an awful lot less than $600. It's certainly something that comes up in the context of advice in Australia. People, they know if they they're going back to their country of origin, this vaccine's on the national program, you can get it for nothing. And for this vaccine, for the visiting friends and relatives group, it may be a relevant consideration, but I'm not sure what the politics of referring to it in a document would be.
Yes, thank you for that. And we are aware of the SA1414-2 vaccine as well as the chimeric vaccine that are available overseas. I think that's a relevant point, and our travel medicine practitioners often raise that issue about the availability of other cheaper vaccines overseas. I think some of the issue is that that means if the traveller arrives in country, they need to seek out a vaccination. It's then about 2 weeks for sufficient immunity to develop that we could say they've got a protective immune response. So it's certainly a good consideration, and we mention in the MMWR report about the availability of other vaccines, but I think for the US traveller, clearly it's not licensed if they want to have protection before they depart overseas. But yeah, VFR travellers are, as you note, an important group, and often may not even access healthcare at all, or it may just be a primary practitioner.
Dr. Whitley-Williams?
Pat Whitley-Williams, NMA. So along those same lines, there may be a U.S. traveler who did receive a vaccination for the first time, then goes to a country and stays for 1 year, 2 years. Let's say they don't come back to the States after that 1 year. We need to make a comment about lack of knowledge about interchangeability with other vaccines that might be available. Just a caveat, or just to make some mention, unless there is knowledge about interchangeability of the two vaccines.
Right. So in our MMWR document, we note that there's other vaccines available overseas, but they haven't been licensed or reviewed in the US, so we can't specifically mention anything in relation to that. But again, it's a good point, and there is some information, but limited information.
Great. It doesn't look like we have any other questions or comments. So thank you guys so much. We're looking forward to bringing this to a conclusion in February. It's been a very long but very impressive road. Can we move on to the next section, please? We have Dr. Stevens, who's the work group chair for the anthrax work group.
Good afternoon. I'm David Stevens. It's my pleasure to introduce the anthrax session for this afternoon. These are the members of the anthrax working group, including Dr. Atmar and Dr. Fry, who are both here, and we're led by Dr. Willie Bauer, who leads us effectively in terms of our deliberations, often energetic deliberations, I might add. This working group was reconvened in March of 2007 to reconsider issues regarding the anthrax vaccine, the U.S. government stockpiles, anthrax vaccine and antitoxin for post-exposure prophylaxis, PEP, should there be a widespread release of Bacillus anthracis spores. The work group has considered the body of evidence since the last review in 2010 for policy changes to optimize the use of anthrax vaccine for post-exposure prophylaxis following a widespread release of Bacillus anthracis spores. And the work group is also reviewing the body of evidence for the use of anthrax antitoxin for PEP when no effective antimicrobials are available. Today's session will focus both informational presentations on a new anthrax vaccine by Emergent, AV7909 for post-exposure prophylaxis, and also an informational session on anthrax antitoxin for post-exposure prophylaxis. And I believe Emergent is going to present the presentation.
Thank you so much. So I'm calling up Dr. Paul-Andre Delam and Mr. Jeff Shearer. I hope I said your names correctly.
Thank you very much. So let's move to the next slide. as was just said, this is really to bring up the audience with the current status of the development of this new vaccine. We're going to review where we are in the timeline until approval, a summary of completed study to give you a background on that. My colleague, Jeff Woodare, will then discuss the basis for the primary endpoint in the upcoming trials, which are planned. There's a phase 3 and a phase 2 clinical trial that we will review shortly. So, that's me, Paul-André Delame, the Vice President of Clinical Development at Emergent. For the background, AV79 is a vaccine, anthrax vaccine absorbed drug substance with an adjuvant CpG 7909. It is developed for post-exposure. It's to be administered IM in 0.5 milliliter dose and two injections at 2 weeks interval. The timeline of where we are planning to submit for an emergency use authorization later this year. It's scheduled for December, and expect to have a BLA filed in 2022. Non-clinical data have been ongoing. Here are the 2 clinical studies that I have just discussed, and obviously we are working hard on making manufacturing happen on time. In terms of current experience with the vaccines, we have performed 3 trials between 2005 and 2015 for a total of exposed subjects of 244. Overall, those trials have shown that the AV7909 as compared to Biotrax, which is the same product without the adjuvant, is comparatively safe and has a better and faster response, in immune response. As you can see here on this graph, the arrow here on the left shows the peak of AV7909, which happens just a bit before day 28, compared to the peak of Biotrax, which happened quite later, 2 weeks later, and is actually lower. So when we look at those studies together, the profile of AV7909 suggests that there is a faster, stronger response. To those are similarly efficacious at day 63. You see here, coming back on this slide, that after that day 42, the 2 slopes decline and are essentially superposable. And in terms of safety profile, they are very comparable, if not identical. I will now give the floor to my colleague, Jeff Shearer, who is going to discuss the rationale for the endpoints for the upcoming phase 3 trial. Jeff?
So a little tag team here. Good afternoon, everyone. As Paul-Andre mentioned, I'll be describing non-clinical studies conducted to support the immunogenicity endpoints for the upcoming AV7909 phase 3 clinical trial. Oops, one way. So the goals of the non-clinical efficacy studies were to determine circulating neutralizing toxin levels in terms of TNA NF50s associated with 70% survival following inhalational exposure to a lethal level of anthrax spores. The proposed phase 3 clinical study immunogenicity endpoints are supported by a combination of both Biothrax and AV7909 efficacy studies. Because Biothrax has been licensed for the post-exposure prophylaxis indication, and because of the similarity of the products, it was considered prudent to determine whether AV7909 given on the proposed schedule is not inferior to Biothrax given on the licensed post-exposure prophylaxis schedule. The Biothrax vaccine was evaluated in well-established rabbit and non-human primate models, and the total of 6 non-clinical efficacy studies of the AV7909 were conducted, 3 studies in guinea pigs and 3 in non-human primates. The reason we were using guinea pigs for the AV7909 is because of the reduced responsiveness of rabbits to the CpG 7909 adjuvant. In the Biothrax studies, animals were vaccinated on day 0 and 28, and then challenged on day 70. For AV7909, we evaluated 2 vaccinations scheduled, an accelerated schedule on day 0 and 14, and also the day 0 and 28 schedule. For both studies, the animals were vaccinated with various dilutions of vaccine to produce a stratified immune response prior to challenge. Separate groups of animals were evaluated in or were challenged in the AV7909 studies. One group was challenged on day 28 and one on day 70. For any of the deaths that occurred following vaccination and challenge were confirmed to be deaths due to anthrax based on both bacteremia and or histopathology. So this figure shows the correlation analysis used to determine the relationship between survival and circulating neutralizing antibody titers prior to challenge. And this was the study that was conducted, or this is the data from the rabbit licensure enabling study, and it showed that a TNA value of 0.56 was associated with protection of rabbits in that study. In the Biothrax non-human primate study, the TNA threshold value of protection was determined to be 0.294, and this is the value that will be used to support the proposed AV7909 phase 3 non-inferiority endpoint. These figures show the TNA NF50 threshold values associated with 70% protection in NHPs vaccinated with AV7909 according to the 0 and 14-day accelerated schedule. The 0.151 TNA NF50 threshold value on day 28 will be used to support a secondary immunogenicity endpoint for protection early protection in the phase 3 clinical trial. We did a meta-analysis where we combined all the vaccination and challenge survival data, TNA data and survival data from all the animals that were either vaccinated with Biothrax or AV7909. Did this meta-analysis, and the conclusion from the study was that because of the similarity in the threshold of protection among the 2 vaccines, that the 0.29 value from the Biothrax study represented a conservative endpoint for bridging, the bridging endpoint for the phase 3 clinical trial. So just so you're aware, we've submitted all these data to the FDA, and we just recently received feedback from them indicating that no additional non-clinical efficacy studies will be required to support the phase 3 endpoints. So, So in summary, the TNA 50 values derived from non-clinical studies supporting the Biothrax licensure for the post-exposure prophylaxis indication will be used for the co-primary immunogenicity endpoints for AV7909 phase 3 clinical trial, and the 0.15 NF50 threshold derived from NHPs vaccinated with AV79 according to this 0 and 14-day accelerated schedule will be used for the secondary endpoint in the phase 3 trial. So I'll turn it back over to Paul Andre.
So the first study that I will shortly describe is a phase 3 randomized double-blind trial to verify lot consistency, immunogenicity, and safety of AV7909 in the indication of post-exposure prophylaxis in healthy adults. with the title already summarized the study purpose, the primary objective is to show that those three pre-production quality lots, to spell out PPQ, achieve a similar titer at day 64. A total of 3,850 subjects will be enrolled in approximately 40 sites, probably a little more. We have 5 groups, one for each of the lots of AV7909, and one as a control for Biotrax, with the numbers shown for each group on the slide. The co-primary endpoint, and co-primary is co-primary, they both have to be met for the study to conclude, I would say, positively for the product, is equivalent immunogenicity across the 3 consecutive AV7909 lots, as shown by a 95% confidence interval for the ratio of geometric means. Apologies for reading, but it's hard to summarize, of TNA and a 50 at day 64. So it's a relatively long-term follow-up for that. And the 95% interval has to be between 0.5 and 2.0. The co-primary endpoint is to show that at the end of the day 64 period, the two-sided 95% interval has to be greater than 40% for the proportion of AV7909 in each of the 3 lots, achieving a TNA NF50 greater than 0.656. At day 64, we will also check immunogenicity, again with the endpoint as defined there. The non-inferiority endpoint, which is the second bullet at day 64, is going to compare the proportion of subjects protected by Biotrax with those protected by AV7909, and the difference has to be greater than minus 15, the minus being not a typo, but in the event Biotrax happens to fare better, it could actually end up being a negative difference. There are additional immunogenicity endpoints and the usual safety endpoint in this type of trials: adverse events, serious adverse events of special interests, etc. The second trial is aimed at assessing the interaction between vaccination by AV7909 and the kinetics of 2 antibiotics that are stockpiled for treatment, i.e. ciprofloxacin and doxycycline, and vice versa, whether they have an effect on the immunogenicity generated by AV7909. It's a study of a total of 210 subjects. 70 will receive AV7909 alone. And then there are 4 other groups. The only difference is that you have 2 groups with Cipro, 2 groups with Doxy. And among those 2 groups, 40 will have a PK for ciprofloxacin or for Doxy, and the other ones will not have to endure the pain of going through a PK sampling. The primary endpoints are the area under the curves for either ciprofloxacin or doxycycline. And again, the safety endpoints are the classic safety endpoints that this type of trial requires. Pharmacokinetics and immunogenicity of AV7909 will also be assessed. So that's a summary of where we are at Emergent with this plan. And if there are any questions, Jeff and I can at least attempt to answer.
Thanks for that presentation. I'm a little confused because on slide 7, it looks like you get a good antibody response when you check humans' blood for the antibody that you're looking for. But then you go over to the animal models in slide 16, and then it's not protecting them from dying as well as it should. Because you're not getting a TNA above 0.56 or whatever your usual cutoff is. That says that it's a good response. Am I not understanding this correctly, or what?
I'm sorry. We're saying that the threshold associated with protection in AV7909 is actually lower. That's the point that 70% of the animals are protected at 0.15, or they're actually all, for AV7909, all the threshold values associated with protection are lower than 0.56.
Oh, so it's just in rabbits where it's 0.56.
Right.
Yeah. Okay, so I missed that part. So it's in non-human primates, the cutoff is 0.15?
The non-human primate is 0.15. That's the threshold of protection.
And the cutoff in guinea pigs is what?
I'm sorry.
And the cutoff in guinea pigs is what level?
The cutoff in guinea pigs it's similar for both schedules. It's around the level of detection 0.063, and at the 0 and 28 and 0.62 for 0 and 14 and 0.08 for.
Okay, I'm sorry, I just didn't understand that. Thanks.
Depending on the time point, depending on whether they were challenged on day 28 or 70, it's a little bit higher on day 70.
Good.
Dr. Stevens?
This is a novel CPG adjuvant, as I understand it. Is that correct? And can you comment on your choice of that particular adjuvant that has been some of the discussion in the committee?
The less than elegant answer is I can get back to you on the reason for the choice. The fact of the matter is that I've been involved in this program relatively recently. The choice of CPG, this being said, CPG was chosen, I guess, because it is known and safe. But why not aluminum, for instance? This I do not know.
Dr. Messonnier?
Just to follow that up, this vaccine is meant to be a vaccine as a contingency plan in the setting of an anthrax attack.
Yep.
And so understanding all the constraints about that and the ethical considerations around use of a vaccine in a population that's not at risk. I just want to ask a little bit about children. So I expect that you don't plan to do studies in children as they're not at risk for anthrax unless there's an event. But in the setting of a large-scale event, this committee will be faced with the real need to make recommendations for use of an anthrax vaccine in children. So with that as a setting, can you tell us what we know about the safety of CPG in children?
It's not really the object of the program right now. This is developed for stockpiling. And I would say as a rule, we'd like to know how it works and how it fares in adults before exposing children. This being said, it's a very valid point and a real concern should there be massive exposure for this type of attack. So thank you.
Dr. Atmar?
Just a clarification on your lot-to-lot consistency study. The regimen's going to be 0 and 14 days?
Correct.
Okay. And whereas the biothrax control will be 0 and 28?
Yes.
Thank you.
Great, thank you. Are there any more questions? Thank you so much for that presentation, and we'll now call up Dr. Bauer.
Thank you.
Okay, thank you, everyone. So I just wanted to point out that if everything goes well with the EUA and that gets approved, that we may be bringing this back to the ACIP in February for a vote on actually using the vaccine in an emergency. But it's sort of contingency on how the EUA goes through the FDA. But now I'm here to talk about anthrax antitoxin for post-exposure prophylaxis. So here's an outline for today's presentation. First, I will give an overview of the anthrax antitoxins. I will then go over the studies reviewed by the work group on anthrax antitoxin used for post-exposure prophylaxis, or PEP. These include studies on antitoxin for PEP and survival in animal models, anthrax antitoxin effect on acquired protective immunity after exposure to aerosolized Bacillus anthracis spores, and effect on immunogenicity when antitoxin is co-administered with anthrax vaccine absorbed, or AVA. Then I will go over the work group's discussion on anthrax, or on antitoxin for PEP, and end with the proposed guidance on anthrax antitoxin use for PEP. First, I would like to start with a brief overview of anthrax pathogenesis and how anthrax antitoxins work to prevent disease. Bacillus anthracis produces protective antigen that binds to anthrax toxin receptors on the cell's surface and forms a heptamer. So you can see there that it binds there and forms a heptamer there. Then Bacillus anthracis also produces two toxins that are essential for the development of anthrax disease. These two protein exotoxins, lethal factor and edema factor, bind to the protective antigen heptamer complex there, and are taken into the cell where the lethal factor and the edema factor are released into the cytoplasm. Once released into the cytoplasm, lethal factor and edema factor inhibit bacterial clearance by phagocytes and help bacilli to escape from macrophages, thus allowing overwhelming bacteremia to occur. So anthrax antitoxin binds to protective antigen, which blocks movement of the toxins into the cell. And so this stops the ability of the toxin to enter the cells where they would exert their effects. However, it's important to note that any toxin that is already, what did I do? There we go. Any toxin that's already in the cell, the antitoxin has no effect on the toxin that's already in the cells. So there are three antitoxins licensed for treatment of inhalation anthrax. Raxibacumab, which I will refer to as Roxy during the rest of the presentation, was first approved in 2012. It is a monoclonal antibody. Anthrax immune globulin intravenous, or AIG-IV, was approved in 2015. It is a purified human IgG polyclonal antibody collected from individuals immunized with AVA. And oblitotoximab, which I will refer to as Oblit during the rest of the presentation, was most recently approved in 2016, and like Roxy, it is a monoclonal antibody. As we saw in the previous slides, they all work by binding the protective antigen produced by Bacillus anthracis and prevents the incorporation of toxin into the cells. All three antitoxins have an indication for treatment of inhalation anthrax in adults and pediatric patients in combination with appropriate antimicrobials. The monoclonal antitoxins, Oblit and Roxy, also have an indication for post-exposure prophylaxis following exposure to aerosolized Bacillus anthracis spores when alternative therapies are not available or not appropriate. AIG IV does not have an indication for post-exposure prophylaxis use. This presentation will focus mainly on the monoclonal antibodies for post-exposure prophylaxis. Of note, these antitoxins are all approved for their listed indications under the animal rule, which means that their effectiveness for anthrax treatment and PAP is based solely on efficacy studies conducted in inhalation anthrax animal models. Now I will review the data presented to the work group on antitoxin use for post-exposure prophylaxis. To show the effect of administration of Roxy on post-exposure prophylaxis, rabbits were exposed to a spore challenge of 100 LD50s and administered Roxy at 40 milligrams per kilogram at 0, 12, 24, and 36 hours after exposure. 100% survived when Roxy was given at time of exposure or 12 hours post-exposure. If administration was delayed to 24 hours or 36 hours post-exposure, survival dropped to 50 and 42% respectively. To show the effect of administration of Oblit for post-exposure prophylaxis, a similar study was done. Non-human primates were exposed to 200 LD50s of Bacillus anthracis spores and administered Oblit at 16 milligrams per kilogram at 18, 24, and 36 hours post-exposure. 100% survived when Oblit was given at 18 hours post-exposure, 83% at 24 hours post-exposure, and 50% at 36 hours post-exposure. To test the impact of Roxy administration on the development of a protective immune response, 21 non-human primates that had received Roxy following a Bacillus anthracis spore challenge of 100 LD50s in a dose-ranging efficacy study and survived were used for a re-challenge study. In this study, these animals were re-challenged with 100 LD50 of Bacillus anthracis spores 11 months after the dose-ranging study and received no PEP. 100% of the animals survived compared to 0% of controls. A similar study was conducted with Oblit to determine if it interfered with the development of a protective immune response. In phase 1 of the study, rabbits were challenged with 200 LD50s of aerosolized Bacillus anthracis spores and treated with either Oblit, levofloxacin, or a combination of the two. In phase 2, the 13 surviving animals were re-challenged 9 months later with another 200 LD50 dose. All survived compared to none of the controls. These two studies suggest that these antitoxins do not interfere with the development of a protective immune response. The co-administration of AVA and AVIG has been shown to significantly reduce the development of a protective immune response to AVA in a rabbit model. There was concern that co-administration of the monoclonal antitoxins could also blunt the immune response to AVA. To determine if Roxy had an effect on the immunogenicity of AVA when co-administered, a study was conducted in human volunteers. One group received AVA at days 1, 15, and 29, while another group received the same AVA vaccine schedule started immediately after administration of a single intravenous 40 milligram per kilogram dose of Roxy. This study demonstrated that when a single dose of Roxy was co-administered with the first of 3 doses of AVA, the immune response in humans at day 29 was non-inferior to that of AVA given on its normal schedule. The study also showed that the percentage achieving a 4-fold or greater increase in toxin neutralizing activity titer from baseline at week 26 after the first AVA dose was not different between the two groups. The FDA did not request this information as part of the approval process for OBLET, so we do not have data on the effect of OBLET and AVA co-administration. Now I will summarize the anthrax work group's discussion on the anthrax antitoxins for post-exposure prophylaxis. The anthrax monoclonal antitoxins, OBLET and Roxy, have an indication for use as post-exposure prophylaxis for inhalation anthrax when alternative therapies are not available or not appropriate. Situations where alternative therapies are not available or not appropriate could include the organism is resistant to all available post-exposure prophylaxis antimicrobials or supplies of antimicrobials to which the organism is susceptible are not available. The data show that anthrax antitoxin administered 12 to 18 hours after exposure to aerosolized Bacillus anthracis spores can prevent 90 to 100% of disease in both rabbits and non-human primates. However, survival rates are both concentration and time dependent. Protection drops significantly with increased time to intervention after exposure. In the studies we reviewed, after 36 hours, the survival in rabbits administered the maximum dose of Roxy dropped from 100% at 12 hours to 42% at 36 hours. Likewise, the survival in non-human primates administered the maximum dose of OBLET dropped from 100% at 18 hours to 50% at 36 hours. In animals that survived Bacillus anthracis spore challenge, there was no late emergence of anthrax from residual spores, and surviving animals were resistant to re-challenge up to 11 months later. This suggests that anthrax antitoxins can provide protection until and do not interfere with the development of a protective immune response. However, the animals in these studies all received high-dose spore inoculums. Whether antitoxin used in persons exposed to smaller dose spore inoculums would interfere with the development of an acquired protective immune response is unknown. Data support Roxy co-administration with AVA as part of post-exposure prophylaxis regimen. When a single dose of Roxy was co-administered with the first dose of AVA, the immune response in humans was non-inferior to that of AVA alone. However, no data on immune response are available on co-administered AVA on a dose-sparing vaccine schedule in conjunction with Roxy. In addition, no data on immune response is available for co-administration of AVA in conjunction with OBLET. Since OBLET is a monoclonal antibody that is similar to Roxy, it is not unreasonable to believe that it also would not interfere with the immune response to AVA. However, at this time, we have no data to determine if OBLET would interfere with development of a protective immune response if co-administered with AVA. In contrast to the data on co-administered Roxy and AVA, the co-administration of AVA and AIG-IV, which is a polyclonal antibody, has been shown to significantly reduce the development of a protective immune response in a rabbit model. These are the statements we would like to include in the policy statement regarding use of anthrax antitoxin for post-exposure prophylaxis. In situations where no alternatives for PEP are available after exposure to aerosolized Bacillus anthracis spores, OBLET or Roxy may be considered to help prevent inhalation anthrax. The predicted effectiveness of both these antitoxins are based solely on efficacy studies conducted in animal models of inhalation anthrax. Data indicate that AVA can be co-administered with Roxy for PEP without impacting immunogenicity. Similar data are not currently available on Oblit. It is thus unknown if Oblit would impact the immunogenicity of AVA when co-administered. AIG-IV does not have an indication for PEP use. In contrast to the data for co-administered Roxy and AVA, the co-administration of AVA and AIG-IV has been shown to significantly reduce the development of a protective immune response to AVA in a rabbit model. Now I'd like to see if the ACIP committee has any questions or concerns with this data.
Great, thank you so much. I'll open it up to the committee for questions or comments. Ms. Stinchfield?
Yeah, Patsy Stinchfield, NAPNAP. Thank you. That was very interesting. I'm just reflecting on the practical application of this, having just gone through doing PEP for measles in Minnesota last year, giving MMR within 72 hours and IVIG in 6 days. The clock is always ticking in these situations, and so 36 hours is really short to identify and administer and get everything in order. So one of the practical applications would be to drill this. You'd have to really have everything in order. You'd have to have your protocols in order, your communication in order, your sites where you would administer it, who would administer it, how are they licensed to do it, just the kind of boots on the ground, how would you pull this off is kind of daunting when you look at that 36 hours.
Thanks. Dr. Leary?
Thank you.
Yeah, along those lines, how are they administered? Are these IM or IV or sub-Q?
IV, intravenous.
So that, just to add to that,
Exactly.
that would make it extraordinarily difficult.
So you have to look at this. I mean, we have a lot of classes of antimicrobials that you can use for prophylaxis for anthrax. So this is in the setting where it's a very multi-drug resistant organism. So the chances of this happening are slight, or I'd say even less than slight, but it does have the indication, and there is a potential that it could need to be used in a very multi-drug resistant organism.
Dr. Hunter?
Yeah, just as a ACIP voting member, I've never voted on anything that's been anything besides a vaccine. And are there any other considerations we have for antitoxins or antimicrobials or anything like that?
So can you clarify, Dr. Bauer, what your plan is for incorporating this type of guidance into the recommendations?
Right. So when we write up the MMWR, which will be, the majority of it will be on our vaccines. We just want to include this as an option for post-exposure prophylaxis, so we sort of have everything in one document. That's why we're not asking for a vote. This is just sort of guidance. But it was also nice to have input from the work group on the discussion as well.
So you're not asking for a vote on this. Do we ever get a vote? Do we ever have to vote on these kind of non-vaccine recommendations?
So I think that's a further discussion which we can talk about at not at 5 o'clock in the afternoon,
right,
but
exactly.
in the setting of we can vote on these types of products in the setting of a vaccine as a vaccine preventable disease.
And
I just want to go back to the comments over here and just say that it's clear this is the first step of what is needed in order to even think about implementing these vaccines and antitoxins, and I just don't want you to think that we're dismissing your concerns about the operational complexity. And I think, this conversation is really just starting. These are FDA licensed products, and it'll be on the groups, just like when we talk about clinical guidance. In the same way, this is something that needs to be worked out operationally with the state and local health departments who would be in the setting of having to actually implement this kind of guidance. So, yes, Points well taken.
Dr. Stevens?
Just to follow up, a significant reason for considering the antitoxins was in relationship to AVA administration. Yeah. You saw that the polyclonal does suppress AVA response, whereas at least one of the monoclonals does not. We're uncertain about the other one, but certainly in the context of AVA administration, I think the knowledge of these antitoxins is important.
Okay, Dr. Romero and then Dr. Kimberlin.
Understanding the greater significance of the use of these monoclonal antibodies and the polyclonal antibody. Consideration also for use of live attenuated vaccines in children after receipt of that polyclonal antibody will need to be addressed. But again, I realize why we would be using the polyclonal.
Dr. Kimberlin?
David Kimberlin, AAP Red Book. I think I misheard. Can you say again how the monoclonals are administered, IM versus IV, and how the polyclonal is administered?
They're all administered IV.
OIM option for the monoclonal?
No.
Great. On that note, thank you so much. At this time, it doesn't look like there's any other comments, and we are going to move on to the vaccine supply presentation with Dr. Santoli.
Hello, everybody. I have a brief vaccine supply update for you at the end of the day. I'm going to talk about hepatitis B update, both pediatric and adult, and Shingrix. So for adult hepatitis B vaccine, Merck is not currently distributing its adult hepatitis B vaccine or dialysis formulation, and will not be distributing the vaccines through the end of 2019. Together, GSK and Dynavax, who also make adult hepatitis B vaccines that are licensed in the U.S., have sufficient supply of adult hepatitis B vaccine to address the anticipated gap in Merck's adult hepatitis B vaccine supply. However, preference for a specific presentation, so a vial versus a syringe, may not be met uniformly during this time. In terms of pediatric hepatitis B vaccine, Merck's supply of pediatric hepatitis B vaccines have been constrained since mid-2017, and Merck will continue to direct its limited supply of vaccine to the public sector. They expect that they'll continue to have a limited supply of monovalent hepatitis B vaccine through the middle of Q2 of 2019. GSK is able to continue to cover the supply gap through mid-Q2 2019 using a combination of monovalent and combination vaccines, and preference for a specific presentation may not be met uniformly during this time. And as we've said earlier, but as an important reminder, the expected monovalent supply during this period is sufficient to cover the birth dose for all children, as well as to provide some second and third doses. And next, I wanted to switch over to Shingrix. As you all are aware, due to high levels of demand for GSK's Shingrix vaccine, GSK has implemented order limits and providers have experienced some shipping delays. This is expected to continue throughout 2018. GSK has increased the number of doses available for the U.S. market in 2018 based on this, and further increases are planned for 2019. And then lastly, just a reminder that we have this web page that summarizes vaccine supply shortages. It's updated regularly, and it'll be updated following this meeting. That's what I was hoping to share. Thank you very much.
Thank you so much. Dr. Hunter?
Just to confirm, the hepatitis A adult shortage is over.
So that's a great question. It's true, the very tightness of the constrained supply that we experienced last year is behind us. We are still, however, in the public sector, we are controlling the ordering of that just to make sure that we can continue to support routine vaccination and respond to outbreaks. So we're doing some watchful waiting and a little more controlling. But with that having been said, we're able to meet the demand and the need for the vaccine at this time. Thank you.
Jeannie,
Nancy?
just in case it's not clear, can you make a comment about the flu vaccine availability?
Okay, sure. So at this point in the season, I think people know that we track national flu vaccine distribution. And as of the last report out that we had, which was October 19th, there were more than 125 to 130 million doses distributed. And I apologize that I do not have that off the top of my head. I should. However, that's actually more than we've had at that time of year for any of the most recent seasons. That does not mean that everybody has the vaccine they want, where they want it, when they want it, because vaccine continues to ship throughout the season. Sometimes you go somewhere and the supply is not there because they're waiting for an order to come in. I know that's very frustrating, but I think we have no concerns about the overall national supply for flu vaccine this season.
Great, thank you so much. We are at the end of the second day, so it is time for public comment. First day. Oh, shoot.
Yes, we always were.
So it is time. It is time for public comment. So I have a list of individuals who have signed up for the public comment during this session. I'm going to list your names, and then I have a couple announcements as you guys are lining up. So the first public comment is from Dr. Paul Offit and Amy Pisano. Second is Susan Corgan. Third is Tia Severino. Fourth is Aaron Olszewski. I apologize for my pronunciation. Next is Teresa Burke, Leanne Johnson, Jocelyn Gallion, and then Jamie Lynn Juarez. And if there's anybody whose name I did not call who would like to make public comment, please line up behind Jamie Lynn Juarez. I just wanted to make a couple announcements. I want to remind everyone to please keep their comments to under 3 minutes. It's really important in order for us to hear everybody, and we'd like to adjourn the meeting at 5:30 today. And I did just want to remind everybody what I said after lunch, which is that the World Polio event is going on outside the auditorium, and we ask that you kindly respect their event as you are departing. Thank you. So, Dr. Offit and Ms. Pisano.
Dr. Offit wanted me to go first. So I'm Amy Pisani with Every Child by Two. In November, we'll be known as Vaccinate Your Family. But I am really here on behalf of, I'd say, the whole vaccine community, and especially the National Meningitis Association. We wanted to remember a lovely, lovely woman who passed away last month named Lynn Bozoff. Many of us are wearing a butterfly pin today to remember NMA, and especially Lynn. So if anyone would like to have one of those pins, please come and see me. The folks at NMA are actually at a retreat today talking about their future, and they have a wonderful president that is going to continue with them. So I know they'll be staying strong. Lynn was a constant here at ACIP, and she often brought parents of that had lost their children to meningitis to talk to us, and she often brought survivors. Lynn's son, Evan, was a junior in college when he contracted meningitis, meningococcal meningitis, and that was in 1998. He died 26 days later after suffering amputations of all of his limbs. And in 2002, Lynn founded NMA along with five mothers altogether who had also not heard of meningococcal vaccination. She and her husband Alan and these many families wanted us to know what it was, and I didn't even know back then. I started myself in 98 with Every Child by Two. I'd like to say that because of people like Lynn and Frankie Milley, we know what the word meningococcal is, and we can say meningitis, and I think parents around the country know that word because of them. Now, because of NMA, there's awareness of ACWY vaccine. We now have a vaccine for all preteens and teens, the recommendation, and for others at risk. Many states now require the vaccine for all adolescents, and the rates are increasing steadily. And also, we all have a wonderful vaccine now for meningococcal B. And just a little anecdote, I never got to tell Lynn how this played out, but I went to my pediatrician with my son, who was 16, and they gave him the ACWY, and I said, when is he getting the B vaccine? And they said to me, let's see what college he gets into and we'll decide then if he needs it. And I said, no, I don't think so. And they said, well, then you're going to have to go and get the vaccine at the pharmacist, bring it back to us, and we will give him the vaccine. And so I went to Lynn and I said, what should I do? And she gave me everything I need to do to educate them. And I'm proud to say that they are now giving vaccine to all kids in the practice, and that's because of Lynn. So Paul wanted to say a little bit more.
So I think the best way for me to remember Lynn is to read a few sentences from something that her surviving son Ryan wrote and posted to a number of us. Ryan wrote, quote, "I can never put into words the impact a mother can have on a son. My mother was such a sweet, warm, innocent person who always made me feel loved. I've always been amazed at how she turned the inconceivable tragedy of losing a son, my only sibling, to bacterial meningitis and became an endless advocate for meningitis prevention. She started the National Meningitis Association with several other parents who lost children or their children were disabled by meningitis. She took it upon herself to be an ambassador for meningitis prevention and awareness. This became her passion in life, to prevent other families from having to suffer this dreaded disease. Part of my peace during this difficult time is knowing that she is no longer suffering, and she will be reunited with my brother, her other son, Evan. My mom and I used to always say to each other, I love you to infinity, and she will always be in my heart and loved infinitely." Ryan Bozoff.
Thank you both so much. Next, we have Ms. Susan Corrigan.
Hi, my name is Susan Corrigan. I'm on the board of directors for Informed Choice Washington. I live in Seattle. I have a speech written here. But after sitting here today listening to all of you and watching these presentations, I would just like to ask a couple questions, if that's okay. Is that okay? One is, you've heard a bunch of people talk about that are in the vaccine risk-aware community. And I'm just curious, when you're listening to us speak, are you listening? Are you hearing our stories? Do you understand that we watched our children go through vaccine injury, and children like mine, his pediatrician's the one that called it because it happened in the doctor's office? Do you guys understand that even though this may be happening to a small subset of individuals, that it's still happening? And to me, it's my only child, that's my whole world. Are you guys listening?
So I just wanted to clarify that we don't respond during the public comment process, but you can go ahead and continue, please.
Okay. I guess I'll just end with this then. My son has a verifiable vaccine injury. It is real, it is possible, it's been proven. I do not wish this on anybody. I wish for all children, I'm just like you guys, I want all kids to be healthy and safe and protected in any ways that we can. And I would just hope that we would all work together and make the effort to start looking at some of the genetic testing for susceptibility. Like my son has mitochondrial disorder and methylation disorders. And we've been told on numerous occasions now that that is a main part of what led to his vaccine reaction. And I would just ask that you guys would consider that evidence and read the book, How to End the Autism Epidemic. If you have not done that, by J.B. Hanley, there are 3 depositions in there that I think would be of interest to you. Thank you.
Thank you so much. Next, we have Ms. Tia Severino.
Hello, my name is Tia Severino, and I am what some might call an anti-vaxxer. That term is not representative of who I am. If I had been truly anti-vaccine, I would not have taken my daughter to get all of her vaccines. I would not have vaccinated my son. When I was here in June, I introduced you to him, and I told you that he only had 4 single-dose vaccines. I firmly believe that those vaccines injured him and caused his autism. My daughter, now 28, was recently diagnosed with Asperger's. I take issue with the theory that the rise in autism is due to a better diagnosis. Children who have autism are frequently not diagnosed for a variety of reasons, not the least of which being the stigma attached to that label. There were not 1 in 40 autistic males when I was growing up in school. They did not exist. It is not better diagnosis. Truthfully, autism is a label that we give to brain injury with gut involvement. Good friend of mine, James Lyon Weiler, scientist, wrote this book, The Environmental and Genetic Causes of Autism. In this book, he presents the findings of over 1,000 studies. The idea that autism is a genetic disorder, which has always been with us, deserves a closer look. It turns out that many of the genes associated with autism have to do with detoxification. Ultimately, genetics can only account for less than half of the incidence. No single gene is responsible for more than 1% of autism risk. The rest is clearly environmental. This book, very good. I recommend everyone on this panel to read it. There is no such thing as a genetic epidemic. For these genes associated with autism to result in autism, there has to be an environmental toxin exposure. These genetically predisposed individuals, they can't excrete the toxins. Certain toxins are cumulative. They build up over time, and they further damage the brain, gut, and other organs. There are known toxins in vaccines. I don't think there's a single person in this room that can dispute that there are known toxins in vaccines. Now, we're not saying, we acknowledge vaccines are not the only source of toxins, but they are without question the greatest and most widespread exposure. When this fact is taken into consideration, autism is environmental, even when genetics are involved. What steps are you, the Advisory Committee on Immunization Practices at the CDC, doing to ensure that genetic screening is done to find these vulnerable individuals? The answer, as far as I can tell, is none. Nothing seems to be happening regarding pre-screening children prior to vaccines. Maybe that would interfere with vaccine uptake? ACIP is part of CDC. CDC is part of HHS. HHS is tasked by Congress, according to the 1986 National Childhood Vaccine Injury Act, to do the following. 1, perform safety studies. Perform vaccine safety testing to ensure that vaccines are as safe as possible. 2, promote vaccine uptake nationwide to ensure that the population is protected. And 3, submit biannual reports to House and Senate committees detailing actions taken to ensure vaccine safety. A recent lawsuit against HHS revealed that these required reports have never been submitted, which begs the question, where are the vaccine safety studies? Were they done? If they were done, why has 30 years passed with not one single report to Congress, as required by law? Over the past 3 decades, HHS through CDC and this panel's recommendations has aggressively promoted, you have succeeded at aggressively promoting widespread uptake of vaccines. In 1983, the CDC's childhood vaccine schedule included 11 injections of 4 vaccines. As of 2017, the CDC's childhood vaccine schedule includes 56 injections of 30 different vaccines. That is a 5-fold increase without any safety testing on the cumulative effects or the effects of administering multiple vaccines at once, which is routine. Routine. I am not an anti-vaxxer. I am a vaccine risk awareness advocate, and I am part of a growing population that is informed and aware of the truth of the vaccine program. Every time a child or an adult is injured by a vaccine, a potential activist is born. If this committee continues in the way that it has, using manipulated, insufficient, and sometimes fraudulent science to justify adding more and more vaccines to the schedule with zero regard for how these vaccines may injure, disable, or kill, you will continue to see our numbers grow. I sincerely hope that you and your peers in the pharmaceutical, medical, and scientific research fields can start to see us for what we really are: intelligent, informed, fearless, capable of doing our own research. Our numbers include scientists, doctors, nurses, and ordinary folks that had to figure out what happened when a vaccine took our perfectly healthy baby and made him sick, disabled, or dead. There's also those among us who are not injured or affected. Some are raising unvaccinated children, and they're concerned about the trend towards vaccine mandates and what that means for their family. The label anti-vaxxer tries to lump us all together in one group, and it has the effect of discrediting us as unreasonable, anti-science, and pro-disease. The reality is, we have legitimate questions and concerns that are being ignored or dismissed. It is time for the CDC and HHS to do the right thing and find ways to reduce harm. Ensure that vaccines are made safe, something I don't think is possible, by the way. And stop enabling the only industry that not only enjoys immunity from liability when their products harm, they also are the beneficiary of this panel's recommendations. I'm sorry, I am emotional, but you have to understand. I'm dealing with something every single day that you all want to deny exists. You all want to pretend like it's anything but the vaccines. I really appreciate you giving us the opportunity to come here and speak. Thank you very much.
Thank you so much.
Good evening. My name is Teresa Burke. I'm from Michigan. I am deaf, so I may mispronounce some words, so bear with me. I'd like to say that I find it very disturbing that we, as a society, accept the following as the new normal childhood illnesses: cancer, asthma, allergies, ADHD, eczema, childhood bipolar, SIDS, juvenile diabetes, autoimmune disorders, seizures, autism, ASD, speech delays, learning disabilities, head banging, nonverbal children, children past the age of 5 that are still in diapers. We accept these issues simply because these childhood illnesses or neurological disorders are not contagious. A child with HIV can go to school. A child with hepatitis B can go to school, but a child that does not have a hepatitis B vaccine cannot go to school. That is unacceptable. I also find it very disturbing that we are led to believe that vaccines are safe with no possible side effects and that one shot fits all. In 2006, both of my daughters were nearly destroyed by the HPV vaccine. Actually, they were destroyed. Years of sickness with no answers from the doctors because doctors are trained to ignore adverse reactions. I have been studying vaccine ingredients since 2010, and I have yet to find safety studies to prove the multiple vaccines given at one time are safe. According to the CDC recommended vaccine schedule, a 2-month-old will receive 7 vaccines. Then again at 4 months, another 7 vaccines. At 6 months, the child will receive 8 or 9 vaccines, depending on if they get the flu shot. There's no studies to prove these multiple vaccines are safe for these small children, and the children cannot speak to us to tell us their adverse reactions. We all hear that everyone wants to prevent illness and deadly diseases. We want health. But 8 out of 10 of our United States of American children have some type of learning disability or neurological disorders or a serious health condition. The United States of America has the highest rate of vaccinations. We have the highest rate of unhealthy children and the highest rate of autism. 7,213,599 children are currently on psychiatric medication. 125,361 of those children are under the age of 1 year old. Such medication is not even prescribed by a psychiatrist, it is prescribed by a pediatrician. You can find these numbers on CCHR International Mental Health Watchdog website. It's unknown how our Amish community survives without all of these vaccinations. As you continue to add to the already heavy schedule, I ask that you please provide our doctors with the safety studies to prove that all of these vaccines given at one time are safe. Thank you.
Thank you.
Hello, my name is Leanne Johnson. I have two severely autistic little boys. They're 6 and 7. I would have loved to be here this morning, but last night I was up almost all night. I got about 3 hours of sleep because my 7-year-old was having myoclonic seizures all night. So one of us was with him, and my other one was having GI spasms, which is associated with autism. So tonight, when you all lay down, please think of me and my children. If you pray, pray for us, because this is a result of vaccine injury for my family. It is constant. We get maybe 2 to 3 hours a night a week. I constantly have family members, therapists, friends coming in every day. It's a revolving door to help us take care of our 2 children. It is very sad, and it is very, very unfair. So we spread out our vaccines, and I went against my gut because I talked to my doctor, who swore up and down that nothing would happen to my boys. We went in on June 27th, 2016 with my oldest, and he was happy. We did the MCHAT, perfectly advanced child. I agreed to do the DTaP because I was spreading them out. A nurse argued with me. She left the room, sent another person in, and they ended up giving him Pentacel, which we didn't know till a couple of weeks later. Pentacel is a 5-in-1 combo, one of those that he didn't need until he was 4 years old. So that's a whole nother issue in itself. So the next day, little did we know at the time, we thought it was kind of cute and he was being silly, but he was already tiptoeing, flapping his hands. He didn't speak, and he didn't speak again for another 8 months. And all we got out of that was a die. We assumed that was for dada. And he was uncontrollably laughing, and he quit sleeping altogether. So I went back to the doctor and I said, "You said this couldn't happen." And he said, "Oh, I don't do autism." And considering that he said that, I'm going to assume that he never reported it to VAERS. So my youngest baby got one vaccine, the same one that hurt my child overnight, the oldest overnight. And so now we deal with severe autism, speech and language delay, sleep disorder, hyptonia, myoclonic seizures, and we now know that they have the MTHFR mutation gene, which affects methylation and detoxification, as well as mitochondrial disorder. So my pediatrician, when I had told my pediatrician that I had the MTHFR mutation gene, I had just learned about it. And again, I should have walked out the door because he said, "I don't really know what that means, and I don't think it affects your child." He should have read up on that because we could have made some better decisions for my 2 children. So if we had a newborn screening for Mito and MTHFR, or at the very least, we had pediatricians that would listen to parents when they present such important information, we would know which children are the ones that are susceptible to these adverse reactions and even death. My healthy children would have been spared, and they would have been living a happy, healthy, normal life. They wouldn't be at home all the time. They would have friends. They would ride their bikes. They would talk. So I've been a part of the vaccine court, which is a kangaroo court of sorts, if you want to call it that. I had to withdraw because our special master, stating similarities to past cases, felt that it was not needed to go before court. What he failed to realize is that our case is almost exactly like Hannah Polling's case, who did win in vaccine court, and also deals with Mito, but they didn't know until afterwards as well. He also didn't read any of the 25 affidavits that I have from my family and friends who witnessed my kids changing before their eyes. I'm not an anti-vaxxer, I'm an ex-vaxxer. And I reluctantly went along with the doctors and the CDC schedule, and it changed our lives for the worst. Our life is hard and it's miserable, and I would not wish it upon the worst person in the world. While I'm here to share my story, I'm not here to fight for my boys, as they'll never be vaxxed again. I'm here to speak for others who are afraid to share their stories and demand better informed consent than a brightly colored VIS paper so parents can make the best decision for each of their own kids, in such as a cases where they don't listen to the parents. As a citizen of the United States, I would like to see a true vaccine safety study on every vaccine, not one, as well as a vax versus non-vax study. We have the data, we deserve the truth, and all the evidence, whether profitable or not for pharmaceutical companies or patent holders. Furthermore, my last statement is that I'm very perplexed by the revolving door between the CDC employees and pharmaceutical companies. I believe anyone who is voting, recommending, paid to work on vaccine safety, or are working on the schedule should not be able to be paid by pharmaceutical companies who make vaccines, nor should anyone who holds a patent. I believe this is a major conflict of interest, and why wouldn't someone to go ahead and vote for what their product could be used to make money on? So the Gardasil slogan is, I want to be one less. I would give anything for both of my kids to be one less vaccine-injured child.
Jacqueline Gallion, I am with Informed Choice Washington. ACIP must immediately end the reckless and dangerous recommendations that vaccine-exempt students be quarantined from school during outbreaks of infections on the recommended vaccine schedule. The primary reasons are the risk of provoking a self-harm attempt or completed suicide and the total absence of evidence that school quarantine is an effective intervention. You hear in the CDC list suicide as the second leading cause of death in youth aged 10 to 24, only behind accidents. None of the scheduled vaccine infections have been anywhere near the top 10 since before World War II. It is an objective fact that some students will and have responded to disciplinary suspensions and expulsions with attempted and completed suicide efforts. This has forced top to bottom reform of how disciplinary suspensions and expulsions are imposed. There is no functional difference between a disciplinary suspension and expulsion and a vaccine quarantine, except the disciplinary attendance probations are implemented much more rationally and humanely. The disciplined student and their mental status is typically known to the excluding authority. During vaccine school quarantines, large groups of unknown students are thrown out with no pre-screening, no monitoring during quarantine, and no reintroduction support, zero. These students are humiliated, discriminated, and segregated, and their privacy is violated. Where are the studies that show school quarantines and mislabeled school exclusions are safe? There are none. This is a reckless, evidence-free practice. There are no studies that show a school quarantine can interrupt an outbreak. In contrast, there is evidence of outbreaks in 100% vaccinated populations. Between vaccines that do not prevent transmission, primary and secondary vaccine failure, antigen escape, and numerous other factors, school quarantines are incapable of interrupting infectious spreading. The uselessness and danger of school quarantines was terrifyingly brought to focus during the 2016-17 Marshallese mumps outbreak. School quarantines were used on exempt students when public health knew all infections was centered in the fully vaccinated Marshallese. The Marshallese were allowed at school because they were vaccinated, even though it didn't work. They should have been quarantined, leaving the Marshallese in school guaranteed a long-term continuing outbreak. In Spokane, some high school students were quarantined for more than 3 months. This jeopardized their GPAs, their sports, their theater, music, art, and other participations, their college admission qualifications, scholarships, and more. Many parents observed mental decline in their children, and one quarantine student died by suicide. This unneeded and unsupported practice needs to stop today. These children are just as valuable. Thank you.
Thank you. Unfortunately, it is 5:30, and so we have to adjourn the meeting at this time because we have to get the room ready for tomorrow. I apologize. I did say that we had to stop the public comment session at 5:30, but hopefully everybody has had an opportunity to speak at least once at this point. And if not, you have not had an opportunity to speak. I can't hear you, but can you speak tomorrow? I can't.
I'm not going to be here tomorrow, and I flew in from Florida. I really would like to say something.
Okay, then please do.
Okay. So my name, I'm going to just take this. My name, maybe they turned my mic off. Okay. Hello? Okay. My name is Erin Olszewski. I am a registered nurse in 2 states. I came here from Florida, and I'm very happy to have the opportunity to speak to you. I'm also Army Special Operations veteran. I did a 1.5 years in Iraq, so I understand what it means to serve our country. And I feel like that should be your job and your #1 priority as well. So I hope you can listen to this and just really think about everything that's really going on with our children, right? I'm here today not only for myself, but also for all the other parents that can't be here and that are taking care of their vaccine-injured children, because there are a lot. There are a lot of them. And we are waking up to this, and it's a very emotional thing for many people to go through, okay? I was a pro-vaccine nurse, and I immunized my patients until our son, who at 12 months went from smiling, bubbly face to talking, and he completely regressed. He took the vaccine, the MMR vaccine at 12 months, took away our son's bubbly, happy character. And this is when I started researching and I started looking into everything that was really, really happening behind the scenes. And the healthcare department that I work for, and I went in to help people, I realized that I was doing the opposite. We are not helping people. You are not helping people. And I know that maybe you may not agree with this, but we are real people, and this is really, really happening. So our sweet boy regressed after the MMR vaccine that was given to him at 12 months of age. He stopped talking. He stopped eating. He stopped all eye contact. He stopped hugs. He stopped kisses. And our lives turned upside down. We had speech therapists. He underwent several tests. And he's just one. He's mine. But all these moms and all these parents are suffering because you are refusing to really look deep down into your hearts and understand that this is not safe. And there needs to be more done about our safety in the United States. So as a nurse, I thought I could trust my healthcare department. And I just can't. I can't bring myself to really comprehend that the business that I went into, thinking that I'm going to be helping people, we are doing the opposite. And everyone in this room needs to just consider this, and they need to look at it and really, really understand that we are real people, and we're going to be back, and there's going to be more of us, and more of us, and more of us, and we are not going to stop until change happens. So I want to just leave you with this. I didn't risk my life fighting overseas for our country so you can just throw it right back into my face. So I will be back, and I'll see you in February. And between now and then, I hope that each of you reaches deep down into your hearts and you really think about what you're doing to our children. Thank you.
Thank you so much for all of your comments. We will be adjourning at this time. We'll restart tomorrow morning at 8 a.m. And I hope you all have a good night.